Hello everybody, welcome to the BeeGuard podcast. And today we have a new podcast with a very exciting scientist from the Basque University, the University of the Basque Country, is it like that? Mm-hmm. And it's you, Melanie, and you know I have stopped trying to say the last name of the people because it's every time when we make a podcast it's getting embarrassing for me. But you are in the BeeGuard project and you're working on the working package number 4, exactly, and that's about the Holobiont. And already now I can feel that I'm on deep water, so I'm very excited to talk about. Tell us, what is actually the Holobiont?
Thank you, Fleming, for introducing me. So, yes, I work on the VP4 Holobiont work package together with Irache. Mm-hmm. And first of all, to explain to you a little bit better what is the Holobiont, it is, we study the microbiota and the host together. Yes. So we don't look at the bees or at the colony just from the host perspective, from the genetics of the honeybee. We want to incorporate the effect of the microbiota.
Exactly.
And look to it together. Yeah. Because if we're talking, for instance, about breeding, the honeybees, they have longer generation times. For instance, 2 years. Yeah. We can make a progress and select in 2 years. But the microbiota actually allow us to adapt and much quicker. They have much higher generation times. We can be much quicker somehow in selecting, in adapting a healthy microbiota to certain conditions, to certain management types. And so we want to look at this perspective, the honeybee and the microbiota together, the holobiont, and how its impact on resilience.
And this is really, then we get back to the word holistic view on the bees, isn't it? So we are looking on the bees under stress condition and non-stress conditions. That's what you're doing now in this project.
Well, we want to compare, and we have several tasks.
Yeah.
First task is we need to set up some more standard protocols for this. This is exactly. Avant-garde methods that we are developing now for the honeybee for the first time. So we need to set up specific protocols, how we can take the most out of the sample types from the gut. We do dissections of the gut. We need to test specific dissection and extraction kits to get the most of the host DNA and from the microbiota DNA. We also want to get DNA and RNA, so also look into expression profiles. What are the microbiota actually doing in the gut? So this is our first task that we will look at.
Simply make a protocol how to do, because this is
standard methods to develop it for hologenomic research in honeybees.
Exactly,
yes.
Yeah.
A side note, just that I talk about it. So we need to know about the microbiota. We know that they have they are very important for immune defense,
exactly.
for detoxification of xenobiotics. They are also important for nutrition, so they help degrade important nutrients so the honeybees can uptake it. So it's really a crucial part of the honeybee health and resilience system. So sorry, because I didn't mention this in the introduction.
But it's really in my life something that has been overseen over the years. So we have been focusing on other parameters actually. So this is very exciting. So what are you doing more?
So the other tasks in our work package are, on one hand side, we want together with VP1 institutional apiaries that we operate across whole Europe.
The
testing apiaries of VP1. So it's really great that we can take advantage of this setup that we have within BeeGuards with all these partners to have access to all these different bees from all over Europe.
Yes.
So we are already receiving some samples from this year's season, and the second task we will do like a catalog, a reference catalog of all the microbiome across Europe.
Okay, great.
This is the first time that it has been done at that level. We will shotgun sequence this and get really deep insight about the different strains that we have, like a diversity of the microbiome of Europe. In the second step, after the the field experiment has been running in all these countries, we want to compare. Do we see differences based on the management type? So, we test the innovative treatment and the conventional treatment in each location. So we want to see if this leaves signatures in the microbiome.
Exactly, and that's, for me as a practical beekeeper, it's extremely exciting. And that's the stress factors that I talked about.
So we would see.
Does it change?
Well, we will see, Fleming. We will see.
Yeah.
We have some hypotheses. We know already from previous research that the environment is one of the key factors, and we know, for instance, also that a simple diversity metric is not always let's say, honeybees that are stressed, they may have more diverse microbiota, but diverse doesn't mean always good. Okay. It can mean that the honeybee is the microbiota is imbalanced in its so-called dysbiosis, and if it's out of balance, you have more opportunistic bacteria that get in, and this can disturb the homeostasis of the honeybee.
But I really try to transfer it to human people. I mean, when I am in stress, I can feel that maybe my holobiont is not really in balance, and I really just should work on that. And that's really with the practices and the way that we're running beekeeping. Yeah.
Mm-hmm. Yeah, we can, I think, draw some parallels to the humans, of course.
Yeah, we should not, but we can.
Yes.
Yes.
And then we also have a task where we have some more dedicated follow-up that is in Italy and in Croatia, because we not just have one apiary, we have actually two apiaries, and one is more in agricultural intense landscape. Another one is more in a natural or semi-natural environment, more closer to the forests. And so there we also expect to see this difference, what I just mentioned, that the environment plays a...
The pesticides influencing...
Yes. In this study, together with VP5, we'll also, in VP5 actually, the pollen will be measured to see the contamination and diversity of the nutrition, the nutritional landscape. I think Gennaro has already talked about this, having the background. Also pesticide will be measured, and to see more the environmental characterization, and then we can see the influence on the microbiome.
And what do you expect? Do you dare?
That's a tricky question.
That's a question. I'm sorry, you don't have to.
So since we already know from previous research, we think that one of the driving factors will be the environment. So we will for sure see across Europe differences. Let's say north-south temperature. Let's say temperature or climate-related influence. But we hope to see, that's why we will take the second sampling after 2 seasons of the differential treatment method, so innovative and conventional. So since we hope that we can also see some differences in the treatment, in the impact of the treatment on the microbiome, but they will be a little bit more subtle. But the thing is, since we have that many these are all like repetitions in all different places, and if we find a pattern that's coming out, that's stable across everything, it's really it's really a good result.
But I can see on you how excited you are about that. How many samples is going to be analyzed in total?
Oh, don't tell me, Fleming, because they're all the time sending more samples and discussing more samples.
But do we talk about 1,000 or 2,000 or what is it?
No. We are talking about less than 1,000, around 1,000 in total,
but
with all the
that's
tasks. And the problem is not 1,000. That maybe if it would just be a bee and we do standardize everything and that's it. The difficult task is the dissections.
The dissection, exactly.
So actually we do in the lab, we are still optimizing this to make it as easy as possible. We do open... Well, we are actually just right now, since we are developing the standard method, we're testing with cutting the abdomen
Okay.
and getting out the intestine.
Exact, yeah.
Or we also do with a pincette, get the sting and
simply just pull it out.
Just pull it out.
Yeah.
But depending on the status of the samples, if they were frozen, defrozen, or how they were stored, etc., sometimes it breaks, and it's a little bit laborious way.
And then the sample is lost.
Yes. Well, we have many bees from a colony, so one is destroyed, we take the next bee. But the thing is, it's a laborious work, and we don't just take one sample per colony, we take a pool of 10.
Yes.
Now, if we're talking about all the European countries, it's 17 institutional apiaries. We take samples now. We will take samples 2026. Each colony 10 bees. We will take a pool of 10 bees. So it's a lot of dissection.
Yeah. Because I'm so happy and lucky that you gave me also sample tubes. So I actually did sample just before I went down here to beautiful Bologna. And then was very important for you. You have to keep it in the cold chain, Fleming. Why do you have to keep it in the cold chain? Why couldn't I just put it in my bag and bring it to you?
Well, Fleming, of course, you know that the DNA got degraded. Yes. The microbes, they maybe move around. It's we know that, for instance, the honeybee DNA is quite stable. We can transport it for a longer time. We can use it in ethanol, and it is much more stable. But the microbes, we are not so sure, because they are inside. They will maybe not so easily get conserved with other methods, and also because we took over an additional task that we have not accounted for, that will be in collaboration with CNR, with Gennaro, is to do also viral analysis of the samples. Yes. And as you know, the honeybee viruses, they are RNA viruses, you know. Yeah. So this is much more easily degraded. Yes. So for there, it's much more important that we try to maintain the cold chain as good as possible.
So it's very sensible to kind of make the sample, take it home to the beekeeper himself, so to say, at the institute, freeze it, and then you have to, as I had to have it on an airplane, and then I had it on the hotel, and then I could take it to your place. So there's lots of places where I could kind of miss it out, so to say.
Yes. This first year we are also trying or testing different alternative methods.
Great.
So there's some special liquid that's called RNA-later. We're doing some tests with that, how well they preserve the microbiota in parallel with frozen bees, and so we can evaluate if it would be easier for in the field realistic samples together with beekeepers or partners like you that don't have access so easily to -80 freezers and to dry ice, and also considering the cost of shipment with these special couriers, etc.
So great. Is there more happening? I mean, it's a lot happening, and I'm very exciting to see the results when you're getting to that place. Are you doing other tasks, or are we at the end now? We are at the end. But then you have to tell us a little bit about your university. Why is your university the best to do this?
Why is our university the best? So in our research group, which is Applied Genomics and Bioinformatics, we work since a long time on different omics, on how to improve sustainable agriculture. We don't work just on bees. Irache has a strong background in microbiota, microbiome studies, but from a different system. So all her knowledge we can bring into BeeGuard. We collaborated in a previous project, European project, HoloFood, that was on microbiome studies in chicken and salmon, so totally different system, but we have really learned a lot about the methods, about the latest technologies, about the latest bioinformatic pipelines. We have a person that's currently in Copenhagen, at the University of Copenhagen, in a research group, our collaborating research group that is specialized on microbiome. So we have experience to bring this knowledge into bees and to do this within BeeGuards.
I mean, we're very happy that we have you guys in the group, and it's very exciting sight that you are working on here. So we wish you the best, and thanks a lot taking the time to explain us.
Thank you very much, Fleming. It was nice talking to you.